The health of the gut microbiome has emerged as one of the most profound influences on systemic wellness, and recent findings suggest that its impact extends deep into cellular self-renewal through the stimulation of autophagy. Probiotics, beneficial bacteria that populate the gastrointestinal tract, play a surprisingly active role in triggering autophagic pathways, particularly in the lining of the gut where epithelial cells are constantly exposed to toxins, dietary antigens, and mechanical stress. One of the most important ways this occurs is through microbial metabolites like short-chain fatty acids, SCFAs, especially butyrate, which are produced when certain probiotics ferment dietary fibers in the colon. Butyrate is not merely a fuel source for colonocytes, it is also a signaling molecule that upregulates key autophagy-related pathways, especially those involved in intestinal epithelial maintenance and immune tolerance. It activates AMPK and suppresses the mTOR pathway, creating a metabolic environment conducive to autophagy. Within the gut lining, this promotes a regular cycle of cellular repair and renewal, improving the integrity of the intestinal barrier. This barrier, often described as the gatekeeper of the immune system, prevents the leakage of pathogens and inflammatory molecules into the bloodstream, a phenomenon known as leaky gut, which has been associated with chronic inflammation, autoimmune conditions, and metabolic disorders. By supporting autophagy in these local tissues, probiotic-derived metabolites help ensure that aging, damaged, or stressed cells are swiftly removed and replaced by healthy ones, enhancing both resilience and adaptability in the gut ecosystem. This in turn creates a positive feedback loop, as a healthier gut lining supports a more balanced immune response and better nutrient absorption, both critical for systemic homeostasis and long-term metabolic stability. Fermented foods such as kimchi, sauerkraut, yogurt, kefir, and miso offer live cultures that help establish and diversify beneficial gut flora. The microbes in these foods, typically strains of Lactobacillus, Bifidobacterium, and Saccharomyces, not only colonize the gut temporarily, but also interact with resident bacteria to promote SCFA production and suppress pathogenic species. These interactions are essential for modulating autophagy, not just locally in the gut, but potentially systemically, as microbial metabolites circulate through the bloodstream and influence tissues like the liver, brain, and adipose tissue. To maximize the benefits of probiotics for autophagy, it is important to pair them with prebiotic fibers, plant-based carbohydrates that serve as food for beneficial microbes. Foods rich in inulin, resistant starch, and pectin, such as garlic, onions, bananas, oats, and legumes, help increase SCFA output and thus amplify the autophagy supportive environment within the colon. The synergy between prebiotics and probiotics, a combination often referred to as symbiotics, enhances microbial diversity and function, creating a more stable and resilient gut environment. Beyond SCFAs, certain probiotic strains have been shown to influence gene expression directly within epithelial and immune cells, increasing the expression of autophagy-related proteins and anti-inflammatory cytokines. For instance, Lactobacillus rhamnosus GG has been observed to modulate intestinal homeostasis by inducing autophagy in Paneth cells, which play a crucial role in mucosal defense. These bacteria-driven mechanisms highlight the fact that autophagy is not only a cell-intrinsic process, but also one shaped by interspecies dialogue at the microbiome level. The broader implication is that gut health is not just about digestion. It's about cellular communication, immune intelligence, and regenerative capacity throughout the body. By nurturing the microbiome through fermented foods and prebiotic fibers, we support one of the most natural and sustainable pathways to autophagy activation, one that is intimately tied to both daily nourishment and long-term vitality. Rather than relying solely on pharmacological triggers or extreme interventions, leveraging the power of microbes and their metabolic byproducts offers a gentle, holistic strategy for encouraging the body's innate ability to repair, clean, and renew itself from the inside out.